What is translation? Translation is the decoding of mRNA as well as using that information to build a polypeptide. This takes place in ribosomes, and ribosomes are in the cytoplasm. As you see on the right, I have a diagram of the overall translation process. Steps of translation. There are four steps. There's charging of the tRNA, initiation, elongation, and termination. Charging of the tRNA, amino acyl synthetase. For each amino acid, this identifies all tRNAs for specific amino acids. Amino acid carboxyl ends is combined to the 3' end of the tRNA. The 5' untranslated region has the Shine-Delgrano sequence. This is relevant because it assists in the ribosome binding. The 5' UTR is identified by the ribosome and allows the ribosome to initiate and bind. Initiation, otherwise known as the beginning of the process. In this stage, the ribosome interacts with the mRNA and the first tRNA so translation can begin. Although we need three important aspects for this to occur. A ribosome has two parts, the large ribosome and the small ribosome, which are made up of rRNA. mRNA that contains instructions for the certain protein that will be built, and third, the initiator, tRNA, which carries the first amino acid in the protein, which is typically methionine, or MET for short. As you can see on the right, I have another diagram that shows the ribosomal unit, the small one, and the initiator of the tRNA, which combines to the 5' cap to make a complex. Next, the complex will scan to find the start codon, and then it will bind to it, making the initiation complex finalized when the large ribosomal unit forms. Next, we have elongation, known as the middle step of the process. In this stage, amino acids are carried to the ribosome via tRNA, then they are linked together to form a chain. As you can see on the right, once again, we have the next amino acid going into the A, but the mRNA shifts forward one codon, thus moving everything over one, as you can see in the diagram above. Next, we have termination, which is known as the end or the last stage. This is when the finished polypeptide is released to do its job in the cell. Termination occurs when a stop codon in the mRNA enters the A site. The release factor, which are responsible for identifying stop codons, are normally in the P site. 3' UTR, which is located behind the stop codon, plays a critical role in the post-translation mutations. If a synonymous mutation occurs, then the vaccine will not have to be changed due to the amino acid not changing. For example, AUU changing to AUC would not change the amino acid, therefore it will not change. Next, in the non-synonymous mutation occurs in the vaccine would have to change because the amino acid changes. For example, AUU to ACU, the amino acids are different, thus having a new chain.